Well, thank you very much for that generous introduction. And I'd like to thank all the organizers for asking me to come here and tell you about what it is we do for a living, kind of. So the first thing we want to talk about is can we knock the lights down, please? I'm not important. The slides are important. There we go. There we go. That's good. All right. So the title of this talk is really DNA, Not Merely the Secret of Life. So what we're going to do is talk about using DNA's chemical information, because that's why it's important, right? It's a molecule that has information, and we're going to use it to control the structure of matter, putting what we want where we want it as precisely as possible when we want it there. Now, everybody in this room knows that DNA is the genetic material of all the organisms I'm showing here, the plants, the animals, the people. And for the next hour, I really want you to forget that, okay? We're not talking about DNA's role in life. We're talking about what we can do with DNA to make shapes of one sort or another. So, for example, this is the configuration of human bones we're all familiar with. But if you do what the artist Deveau did, and strip the flesh from these bones, well, then the reason for organizing them this way becomes a little weaker. So, for example, this is a chandelier in the cemetery of the Capuchin Monastery in Rome. And it's made of bones, in fact, the bones of the deceased members of that particular religious order. And we can see that it doesn't look anything like the skeletons we were just looking at. So it's just a matter of saying, why not? What else could we do with some natural material? Now, bones are fascinating things, but we don't really work with bones. We work with this molecule, DNA. Now, probably everybody who's been to school for more than about a year or two, anybody who's been through kindergarten, knows the DNA molecule, that it's a double helix. But let me just remind you of a few features you might have forgotten. So the form of DNA that we're looking at here, where the base pairs are perpendicular to the helix axis, we call bDNA. And we should remember that DNA is a nanoscale object. So its helical repeat is about 3.5 nanometers, and its width is about 2 nanometers. And there are about 10 or 10.5 units per helical repeat. Now, the business end of DNA is not the backbone here. The backbone is necessary for the molecule to do what it does, but the real business end is here in the famous Watson-Crick base pairs. A pairing with T by 2 weak interactions we call hydrogen bonds, and G pairing with C that pairs with 3 hydrogen bonds. Now, it's also useful to remember that the 2 double helices actually point in opposite directions. So if you look at this pentagon, it's pointing down, but the one across the way is pointing up. So they're anti-parallel, not parallel to each other. Now, it's useful to know where our historical roots are. This is Alex Rich. He was actually my postdoctoral mentor many, many, many years ago now. And Alex was the one who invented the notion of hybridization. So this is the entirety of the paper where he first introduced that concept. But in fact, the part of involving hybridization is only this little red paragraph that I've indicated. And what it shows is that, what it says there is that he wants to point out that this method of forming a 2-stranded helical molecule by simply mixing 2 substances can be used for a variety of studies. So in his case, he was just using 2 strands of DNA to make it, actually of RNA in this case, to make a double helix. We use more than 2, and that's what makes us, what we do a little bit more interesting, perhaps. So this is the DNA double helix again. And it's really kind of hard to make interesting shapes, interesting structures from something like this that has a linear helix axis. Now, when I say linear, I don't mean linear in the geometrical sense of being in a straight line, because it isn't necessarily. It can be curved and can be even knotted. But the point is that it's just going to be a line. It's linear and unbranched. And that limits what we can make out of it rather severely. So what we've done in order to solve that problem is to go to biology and steal something. That's what the word biocleptic means. And what we've stolen is the notion of reciprocal exchange. That does exist in biology. So here, for instance, is a blue strand and a red strand, and following reciprocal exchange, we have a red-blue or a blue-red strand. And that gives us a whole new set of things we can do. So here is an example of that. Here's a double helix, here's a double helix. And at this point, I'm going to make that, perform that operation. I'm going to get a green strand that starts off on this helix, and then crosses over and goes down to here, and another one that goes the other way. So we have 4 strands in one complex now. Now remember, I was saying that the 2 strands are anti-parallel, so that means there are 2 ways we can do it. We can do it between 2 strands that have the same polarity, or 2 strands that have opposite polarities. So when we do that, then the green strand goes down to here, crosses over, and comes back up, and the next one goes around like that. Now, these 2 molecules are actually the same molecule. They're just in different, what we call conformations. So if I flip this helix end for end, I'd wind up with this structure. Later you'll see that if we do this operation multiple times, we really get different molecules. Now, it's easiest to look at this system more when we draw it like this. So it looks like we have an intersection in a highway. So these are 16 mers of DNA, 4 different strands, 1, 2, 3, and 4. And we've designed the sequences here so that they come together to form this single molecule. And we insist, well, I'm not going to go into the details of the design. I'll just say we can do it, and it's rather easy to do it. So now we've got a branched molecule. This is analogous to an intermediate in the process of genetic recombination. Now, once you can imagine having 4 roads, if you will, coming off of a given intersection, well, you can imagine other kinds of intersections, one with 5 and 6 roads coming out. Excuse me. And Yin-Li Wang in my lab reported that in 1991. And even larger types of road mergers, where we have 8 or even 12 of them. And Xing Wang in the lab reported that about a decade ago. Now, I don't want to talk about either of those structures in great detail, but I do want to talk about this one. In the fall of 1980, so long before this work was reported, I walked over to the campus pub at the university I was in and decided to think about 6-arm junctions. Now, when I walked into the bar, I was thinking of them sort of like this, just a more or less planar object, because it's easy to draw it this way. But while I was having my beer, I think it was my first beer, I suddenly remembered this woodcut by Moritz Escher. And I realized that these fish are just like the 6-arm junction. You start in the middle, you have a head and a tail, a top fin and a bottom fin, a right fin on the fish on the left. Far more, so I realized that these things could be 3-dimensional. Far more important than that is that these fish are organized like the molecules in a molecular crystal. So you have a periodic arrangement front to back, top to bottom, right and left. Now, of course, Escher, when he drew this, was totally in charge of what was going down on the piece of paper. But if I were going to try to emulate this, I would have to do something else. Now, one of the reasons that I was so interested in this arrangement was that at the time I was a crystallographer. That was somebody who makes crystals of molecules, mostly biological molecules in my case, and determines their structures using X-ray scattering, X-ray diffraction. And I was starting the fourth of my 5 probationary years as an assistant professor there, and up to that point I'd managed to crystallize nothing of interest to me or anybody else, and I was facing this kind of fatal progression of no crystals, no crystallography, and then, of course, no crystallographer. So I thought this might actually help me in my attempt to survive. But as I said, Maurice Escher was able to put all the little fish where he wanted to put them, but I didn't have that option. I'm a natural scientist, not an artist. So what I did was I took a page from this wall in Nanjing. So you notice there's a brick here, and there's a lot of writing on the brick. And basically what the brick has are instructions as to how to put the wall together. So it goes to the bottom left of this, the bottom right of this, the upper right of this, and the upper left of that. And it turns out DNA has the same type of thing. We call it sticky-ended cohesion. So if we have 2 double helices where one strand is a little longer than the other strand, in both cases, I've unwound these so we can see the sequences, then if this little sticky end is complementary to this sticky end, these guys will come together and form a single molecular complex. If we want, we can even make it covalent. We don't do as much of that as we used to. The other aspect of this is that we know what the structure is when we bring sticky ends together. So this is a crystal structure done by Hangsha Chu in my laboratory, reported about 20 years ago. And it shows an infinite helix in the horizontal direction, but it's held together by sticky ends. You see the little gap here and here, where the overhang from this side and the overhang from this side are coming together, and then they end. And as a consequence, not only do we know that there's affinity between these 2 molecules, there are lots of kinds of affinity interactions. We actually know what the structure is at the end. That's very powerful. So in order to sort of introduce the whole notion of structural DNA nanotech, we're going to combine these 2 ideas, branched DNA and sticky ends. So X can pair with X prime, its complement, Y with its complement, Y prime, and we could make this little quadrilateral. But you notice when we make the quadrilateral, say corresponding to an object, we've got a whole bunch of sticky ends left over on the outside, so we can make a lattice. And we can also make machines, and I'll show you that a little later. So this is a quote from George Bernard Shaw about 100 years ago or so, from Back to Methuselah, one of his best-known plays. And what it says is, you know, this is the serpent in some origin myth. And what it says is, you see things and you say, why? But I dream things that never were. And I say, why not? And that's kind of where we're at here with DNA nano. There's almost no limit to what we can do. So for our lab, the applications are things like macromolecular crystallization, which might lead to drug design, nanoelectronics organization, we're starting to work on that seriously now, DNA-based computation, which is aperiodic, and control of polymer materials composition. I won't talk much about that today. In terms of motion, nanorobotics and nanofabrication, these are things that we work on. I'll show you a little of that. And biomimesis, where we've actually done this now, but I haven't got time to talk about it, doing self-replication, selection, and evolution. We haven't done the evolution yet, but we have done the selection. So let me just, most of you are probably unfamiliar with crystallography. It's a really sophisticated physics-based analytical technique. Most people like myself don't even bother having X-ray units in their laboratories anymore. We go off to a synchrotron to collect our data. or maybe an XFEL source. We have very sophisticated data collection devices, basically electronic pieces of film, so we get quantitation immediately. And we have amazing computer graphics so we can see what we're doing. But in the end, it all depends on the crystallization experiment. You can't do crystallography if you don't have crystals. And how does that work? Well, you guess your conditions, you set up your crystals. Those who incline might go through the middle step, and then you look to see if you have crystals. If yes, okay, great, you can go do crystallography. If not, well, you can either go through the inner loop if you want to, or go back through the outer loop, but basically there's no feedback from failure here to knowing what to do over here. If you go to my web page, you'll see that, in fact, this is the second version of this slide, because in the first version, I forgot that this branch of the loop even existed. So our suggestion is to do something like this. The magenta here represents DNA double helices. These things sticking out are sticky ends. And basically, we've got a box here that's hosting some kind of macromolecule, and we're hoping to just tack the boxes together, and everything will then be crystalline. In the same sense, we can organize nanoelectronic components this way. Here's a branch junction, here's a branch junction. Pendant from it is something that can behave as a piece of electronic circuitry, and we can bring these guys together to organize them using the organizational and architectural properties of DNA. And we're working on that as we speak. Why DNA? Well, it's my propaganda slide, and I'm not really going to subject you to all of it. Let me just say that nucleic acid sequences can be programmed and synthesized readily. That's a change from back in 1980, leading to information-based structural and sometimes dynamic chemistry. I won't talk about the rest of it. So, for example, the very first things we made back in the 90s, early 90s, were polyhedral catenanes, things that were hung together but weren't necessarily well-shaped. So a cube-like molecule and a truncated octahedron were built by Younghui Chen and Yuan Tsang, respectively. So this is the cube-like molecule. This was the first thing ever made with DNA nanotech. That wasn't sort of trivial. And here you can see that each of the faces corresponds to a cyclic single strand. So the red strand corresponds to the front face, and it's linked twice because these edges are 2 turns long to the green, the cyan, the magenta, and the dark blue strands that flank it, only indirectly linked to the yellow strands at the rear. A few years later, Yuan Tsang built this truncated octahedron. That was, it's a 14 catenane rather than a 6 hexacatenane. But this really isn't structure, this is just control of topology, which is much easier. So then we needed to find some way to do this. And this is an example of how we proceeded. The problem is that we had to solve was that if you have lattice design components, they have to have predictable interactions, and they have to have predictable local product structures. Well, we talked about that with sticky ends, but they also have to have structural integrity. That's a fancy way of saying they have to be stiff. And the simple branch junction is not badly modeled by this drawing of or a photograph of a marshmallow that's been impaled by uncooked rotini. So rotini are double helical pasta. And they're impaling this marshmallow. So they're, in fact, quite stiff, but the angles between them are not so well defined because the marshmallow is quite floppy. So in order to solve that problem, we went back to our motif generation protocol, and now we're going to do 2 reciprocal exchanges, either above the line with strand polarities identical or with opposite ones below. And we can make a molecule that has 2 double helices, we call it a DX, or we can keep going as long as we want. We can add 1 more helix and call it a TX. And you notice these are now different. I mean, here I have 2 green helical strands, 2 red strands, and then a blue one in the middle, whereas here I only have 4 strands, 2 reds and 2 greens. So we get to new topologies if we do this. And the very first thing we did with this system was to build two-dimensional arrays from the DX molecule. This was work done collaboratively with Eric Winfrey of Caltech. Frang Liu and Lisa Wensler in my laboratory did the following experiment. We took a DX molecule and another DX molecule, and you can see here we're representing the sticky ends just geometrically. So this complements that, this complements that, this to there, and this to there, and they look like they should come together to form this lattice. Now, we've also got this extra black domain over here. That represents an extra domain of DNA coming out of the screen at us. So that it should form a stripe-like figure. Now, the dimensions here are 16 nanometers, so we should have a 32 nanometer spacing between whatever stripes we get from these guys. And what we see, what we have here is, in fact, a series of stripes. This is an atomic force micrograph. And their separation is about what we predict. Now, there's a basic rule of microscopy. Doesn't matter whether you're using a dissecting scope, an electron microscope, an atomic force microscope like this, or what have you. But if you go looking for something, you're going to find it. So you have to run controls. So the control that we ran is shown over here, where we took 4 molecules like this and stacked them together. And now we should have those stripes separated by double the distance, about 64 nanometers. Maybe I should switch pointers. I have a feeling this one's getting a little bit weak. And we should have a 64 nanometer spacing. Now, I should also point out here that in all of these constructs, all the helix axes are pointing the same way. But at any rate, here in this control, we see that we do have a 64 roughly nanometer spacing on our stripes. So we're doing what we're supposed to be doing. Now, there's a kind of DNA construct that we didn't think up, but which really interested us, called DNA origami arrays. It's a totally different scale from what we've been discussing. This is work done by Wenyan Yu in particular, also Hong Zheng and Risheng Wang. And DNA origami is a technique invented by Paul Rodamund about a decade or so ago, a little more now. And what it consists of is taking a long cyclic strand. By long, I mean like 7250 or so units, and you make shapes out of it. So here is a design that he put in his paper of a hexagon with 2 right angles in it. And what he does is he folds up the circle into this sort of Christmas tree shape, and then he throws in between 200 and 250 little what are called staple strands. So they hold this thing together in this fashion, and then you get out this shape of DNA. So this is much, much larger than what we've been talking about. So what Paul was able to do is he made this square, he made this rectangle, he made this star, and he even made a smiley face out of DNA origami, as well as a couple of trigonal constructs. The important thing about DNA origami is not that you can make a smiley face. The important thing is that in an area of roughly 9,000 square nanometers, if we're going to restrict ourselves to 2D, we have about 200 addressable points. So for example, here is his schematic, and this is the actual construct that he made. This is the Western Hemisphere that he was able to draw with his patterning, the same way we made the stripes. So here's South America, here's North America, there's Greenland. You can recognize the whole thing. So what happens is when you start working with origami, you say, wow, it's really, really big. Then your imagination runs away with you and say, well, you know, it's not big enough. So we decided we would try to crystallize this stuff into a larger arrangement. So this is an example of how we were doing that. And this follows the same procedure that we used when we made that DX array. So here's these are tiles, all have a little hole in the middle of them. And this one's supposed to pair over here and over there and down there and down there. But what we get is something that's basically one-dimensional, sort of 1¼ dimensions. It's not really a nice 2D array. And what Wenyin Yu recognized was that the helix axes are all pointing this way. And he said, well, why don't I just make something where the helix axes are pointing both ways? So he designed an origami that did that, and it looks like this. And then he made this 2D array, and you can see it's authentically 2D without any problem. And that leads us to what we call Wenyin Yu's rule, which is that DNA arrays in multiple dimensions should contain helix axes that span those dimensions, that point somehow in the directions of those dimensions. So this brings us, would have brought us, to, I've got a problem here. How are we doing? Can somebody help me here? I don't know what went wrong. I must have leaned on it. Oh, there you go. Okay. Go back. Back. There you go. Okay. Thank you. So after my afternoon in the pub in 1980, we finally were able to publish this work in 2009, 29 years. This is work done by Jianping Zheng, Jens Bergtoff from my lab, Yi Chen from Purdue, who was the student of Chengde Mao from Purdue. Chengde was my student. We still collaborate. And Tongwan Rujisha and Pams Constantino from my lab, and Steve Janelle from Argonne. And we used a motif that obeys Wenyin Yu's rule, but it was developed by Chengde, and it's called the Tensegrity triangle. So I wouldn't have called it that, but he did. And so here he's got an over-under, over-under, over-under motif. This gives us in the diffraction pattern the predicted rhombohedral symmetry, gives us the right lengths of edges, spacings for the dots, but the resolution is not so good, about 10 angstroms. So Jianping Zheng in my lab figured out that if we shorten this thing down to 2 turns, and then also symmetrized it so that AG here and AG here and AG there would in fact give us a better crystal, and indeed it did. This is another picture of the motif, just accidental. Chengde took this picture at dawn sometime when he was jogging, and you know immediately that had to be Chengde and not me, because I don't do dawn and I certainly don't do jogging. But you can see here that this thing is has an over-under, over-under, over-under motif, and these cylinders are sort of like the DNA double helices that we have. This one's even worse than the last one. Let me try the third one. This one's probably the red, right? Yeah, but it's on. Okay, so by putting these things just together in a pot, we were able to make macroscopic crystals. These are things you can see with your naked eye. They go anywhere from about 0.2 of a millimeter when they behave properly, all the way up to even a millimeter on occasion. So you can see over here that we have, like, the scale bars here are 200 microns. And you can see that we have large, this is probably 300 or 400 microns from across the diagonals here, and large crystals, and we know where all the atoms are because we put them there. But we had to do this crystal structure, and I'm an honest crystallographer, so we made an iodinated derivative and solved these structures by unbiased methods. And then this is the structure of the triangle. This part's in front of the screen. That's in the plane of the screen. That part over there is behind, where this is in front again, and so forth. It's threefold symmetric. And this shows that, in fact, we have the sticky ends holding the different units together right at that point. This is a schematic that gives you a feeling for the crystal structure, so that you can see that we have the central triangle bonded to 6 other triangles, and we have a red double helix starting in the rear and going to the front, a green double helix starting in the rear and going to the front, and a yellow one starting at the rear and going to the front. So it is a 3-dimensional arrangement. If we select a different group of triangles, then we have something that's a little more meaningful. This is a rhombohedron. Remember, this is rhombohedral symmetry. So we have the rear vertex of the rhombohedron is flanked by this red triangle, and it's connected to these three yellow triangles that are somewhat closer to us. They flank vertices closer to us, and then the green triangles are connected to them, and they flank vertices closer yet. This is an artist drawing, courtesy of David Goodsell, showing the same thing. Here's the rear triangle. These three are further back, and these three are closer to us yet. But now we can see how this whole thing fills space in 3 dimensions. This wasn't a lucky punch. We've been able to, this is an old slide, we've made many more, but at the time we were able to make 2-turn, 3-turn, and 4-turn triangle lattices with and without the symmetry. And this just shows the volume inside the boxes. So that's 200 cubic nanometers and about 550 to 600 and about 1,500 or so for the larger ones. Now, one of the problems is the resolution gets worse as we go up in size, and we're working on that. But the very best one we've actually taken down to about 3 angstroms resolution, which is pretty good. This is a movie that shows you the 4-turn helix because it's the most photogenic. And we can watch it actually forming a rhombohedron for us. And you can see there are 4 of the triangles. And here are 4 more, 3 more. And here's the last one. And you can see the rhombohedron that we're able to form. Now, what you see on the screen, of course, contains 8 triangles, but the real crystals contain about 50 trillion of them. So that's a little bit larger arrangement. So one of the nice things about being able to control what goes into your crystal is that you can choose the number of repeat components in the lattice, work done by Tong Wang, Ruji Shah, Jens Bergthoff, Zhenping, and Xingde Mao. And you can see here's the rhombohedron again, but every edge is flanked by either a red or a green, or a red and a green triangle. And these are chemically different, so we've actually got different chemistry going on there. You can see a little bit better here. It looks like 2 intersecting tetrahedra, but they're not regular tetrahedra because this is a rhombohedron, not a cube. Right. So what Ruji Shah was able to do was to actually control a macroscopic property here, namely the color of the crystals. So he took a pink dye called CY3, and he put it on the A molecule, the B molecule, or both, and he got pink crystals. Took a blue molecule called CY5 and put it there, either on A, B, or both, and got blue crystals. And then in either order, he put the pink one on A and the blue one on B, or vice versa, and he got purple crystals. So we could control a macroscopic property that way. Now, one of the interesting things about DNA is that you can use it to organize other materials. So in this case, we're looking at work done by Jiwen Zhang and Pam Constantino from my lab, Christine Michael from Paul Alvasados' lab, and Rick Keel was involved when he was at Minnesota. So this is that same triangle again, but now it's big. So this is a DX instead of a single DNA duplex. It's 8 turns long, and we're going to do this work in only 2 dimensions. So we're just going to make a planar arrangement. And here we are doing it with 1 tile. Here we are doing it with 2 tiles. And you can see how we're doing this now. We're going to propagate in this direction. We're propagating in this direction. The third direction, we just attach a nanoparticle to the end. And this nanoparticle is attached to a single strand of DNA. That's an inherent component of the motif. So here are the 2 different triangles. You can see they're color-coded. One of them is attached to a 5-nanometer particle, gold, and the other is attached to a 10-nanometer particle. And we should get a checkerboard pattern out of that. And And we do. You can see the checkerboard here in the transmission electron micrograph. Recently, we've finally been able to move into 3D. This work is not even in print yet. Yui Zhao from my lab and is working with Ricky Nemeth and Chris Ackerson from Colorado State, and they've got a little gold particle here on the Tensegrity triangle. And this slide just shows that using scattering, it's really only going to be sensitive to the presence of the gold. We can put them there in 3 different positions. This is, again, symmetric. Here we have the same thing on the top and on the bottom. And then here you can see that in the top we have the gold scattering, and the bottom we have just a different scattering, and you can see they fall in the same place. So now let's move on from just structure to machines, and we're going to add time to our coordinates here. The first device was built by Chengde Mao in the lab back at the end of the last century, and it was based on the following notion. This was a DX, this was a DX, 2 double helices of DNA, and the middle was a bit of DNA that could turn into something known as zDNA, which is left-handed DNA. And it could do that only when cobalt hexamine was added to the solution. When that happened, this domain of DNA would flip to the other side of this long shaft, and we could tell that by fluorescence resonance energy transfer between these 2 dye positions. I didn't ever like that device because it didn't really take advantage of the programmability of DNA. So Hao Yan, shortly thereafter, made a sequence-dependent device using this motif that we call PX, where we're now on the top of the screen. And everywhere there's a point where these guys face each other, we've made a crossover. And over here, it looks like we have a turn and a half of blue double helix wrapped around a turn and a half of red. It's a little more subtle than that. But we have AB and CD down here. If I get rid of these 2 crossovers, then I have AB over here and DC down here. So we flipped it a half turn. And the way we get that to happen in real life is to break the red strand here and put in this green strand, and break the blue strand here and put in this green strand. And these horizontal segments are what are called toe holes, a method invented by Bernie Yerke when he was at Bell. And what we do, they correspond to about 8 units of DNA, 8 nucleotides. And if we throw in the whole complement of these guys, they'll sit down over here on this toe hole and then work their way up to here and pull out the green strand. So this shows the same thing. And we put in the complements, the little black dots are biotins, and we can pull out those greens. We have a naked frame over here. We can add the yellow strands, and now we make the other states. So here's CD, here's DC. And then we can do the same thing with the greens and with the yellows and go back to the green state. And the way how I showed this was by taking these half hexagon trapezoids, connecting them with the device. In the green strand state, they're all parallel. In the yellow strand state, it's a zigzag pattern. And here in the atomic force micrograph, in successive cycles, we have parallel, zigzag, parallel, and zigzag. Banani Chakraborty from my lab was able to turn this into a 3-state system, where this was the rotation I was just telling you about, but you could also take these guys and fold them out to make this little X-shaped molecule and give us 3 states for our money here. The next thing I want to talk about are walkers. This is the first DNA walker. It was biped built and designed by Bill Sherman. And it walks like an inchworm. So this foot is always behind this foot, but you can walk in either direction. And we can watch it. This is the walker, and this is a TX sidewalk that it's walking on. And here you can see in more detail how we lift off the rear toe hold molecule, and we put in a new one that holds it down here. And we can take it in both directions. So given all of that, let's do something a little bit more interesting, which is to combine a bunch of multiple components here. So here's an example of doing that. This is a self-opening umbrella. And the way that works is we have a cloud over here. The rain comes in. It soaks the sponge. It expands. That flips this lever so that the thumb hits the striker wheel, and that lights the candle. When the candle is burning, then the water in the kettle will boil, and when the water boils, the whistle blows. The monkey hears the whistle, it jumps on the trapeze, it swings back and forth. The knife cuts the string, the helium-filled balloon rises, it opens the cage door, and all the little birds attached to all the ribs of the umbrella fly in different directions, and it keeps the guy dry. Now, this is probably more efficient than what we can do at the nanoscale, but we're still working on that. So our components here consist of programmable switches, clock walker, nanoparticles, and DNA origami, and it results in a proximity-based programmable nanoscale assembly line built by Hangzhou Gu, Jizhou, and Xiaoyan Xiao. So this is an assembly line from Detroit in the 1920s, making a car. And what you see are three key components. The most important one is the assembler, the worker who knows what to do at any given station. Then there's a conveyor that moves the growing car from station to station. And then there's the factory in which it's being done. That's the framework. So what we've done here is we've replaced the person with the PX-JX2 device, but fused with the origami, so it's in a cassette, and that's the framework. And then the interstation conveyor is another version of the same trigonal walker, except the Tensegrity triangle, except that it's also the chassis. It's going to get increased in size. So if we look down this row of this schematic, this column of schematic, here we see in schematic what we see over here in full color AFM. So we actually, here we have the system. There's the origami, the little notched rectangle. And there are three stations that have cargoes, and they're all in the off condition at this time. That's a 5 nanometer particle, a pair of fused 5s, and a 10. And in the first step, we're going to turn this one on, and it's going to add to our walker. And then it's going to take a half step and a whole step and wind up under the second position. You can see that over here. And then we'll turn this guy on, and it'll add to him over here. And then we'll take our half step and our whole step and wind up under the third position. And then eventually we'll add it all together, and we'll release our product. So the walker itself has 3 hands for collecting cargo, H1, H2, and H3, and 3 feet for walking, F1, F2, and F3, and a 4th foot to orient it towards the loading stations. The way the transfer works is that when this thing is pointing towards the walker, then towards the triangle, which is the walker, then this bit of DNA attacks over here, and we will form more base pairs, and then it'll just displace these base pairs because there are more base pairs here than here, and we get the transfer to occur. We have programmability. We can make any of 8 products, adding nothing, or just a 5 nanometer particle, or just a 10, or just the pair of 5s or the 10, the pair of 5s and the 5, the 10 and the 5, the pair of 5s and the 10, or all 3, as I showed you in detail. And these are the ends of the walks. These are the 8 products again. And then on the right here is an AFM of the, an electron micrograph of the products. So most recently, we've made a traffic light-like device that works in the 3D crystal, work done by Yudong Hao, Ruji Shah, and Martin Christensen. And this is a building in my neighborhood. It's the Empire State Building, and you can see that we have over here a traffic light, and there are crystals in the lights. That's kind of a construction, of course. But what we've been able to do is we can start off with the crystal in a colorless state, and we've extended the central strand there, so it's long. We can throw in a dye-bearing strand that has the pink dye on it, and when it binds, we get a red crystal. And then we can throw that red strand out and put in the blue strand, and now we've got the blue crystal, and we can go around this cycle quite a few times. This is just crystallography showing particularly on the bottom, that we're pairing there like we're supposed to in both cases. That green stuff means that we've got electron density where we need it. Another thing that we've done is my colleague Jim Canary and his student Xiao Wang, along with Ruji Shah from my lab, have self-assembled 3D polyaniline crystals. This is a semiconductor. And we've connected the polyaniline to pieces of DNA on each end, and they connect to two triangles. And then, so this corresponds to an edge of the octahedron. And normally this is blue, but if we lower the pH, it'll be green. If we oxidize it, it'll be pink. And if we reduce that, it'll be clear. And then these are the crystals. Here it is blue. Here we can see it green as we lower the pH. As we oxidize it, it turns pink. And as we reduce that, it becomes colorless. A final important thing in this direction is an oriented guest in the lattice. Yui Zhao, Ruji Shah, and Jens Berghoff put this together so that this is a triple helix of DNA. But triple helices are fairly common. They're actually also discovered by Alexander Rich. And this was just a chandelier in a motel I was staying in one time. But this is the edge of a three-turn triangle in yellow. And then the electron density, this kind of slug-like looking thing, shows that we have the triplex-forming oligonucleotide binding in the major groove there. Finally, we're going to talk about moving up from the nanoscale to the micron scale. Work done with Matan Ben-Zion and Xiaojin He, who worked with Paul Chaikin's laboratory as well as ours. And the components are another origami. It's a 4-helix origami belt. And it's long. It's about 600 nanometers long. It has little connections that will attach it to a colloid. And then two of these guys attached to two edges of that cross-shaped origami so that we have an L-shaped binder. And so what we have here, here's the colloid. Here's the C, B, and A binding positions. So the green binds to the red over here because that binding position is on the cross-shaped origami. And then if we want, we can put the blue and the green over on these sides, or we can put it on this side, so we can actually make mirror image constructions here. And this is not something that's ever been done before, so we can make the left and the right-handed version of these tetrahedra, and only those. So to summarize what we've done, I guess, can you help me again? Okay, we're back, I guess. Okay. So to summarize, we can make knots and bromion rings. They didn't talk about that. Catenanes I talked about a little bit. From branched DNA with ligation, 2D lattices with tunable features have been made from DNA origami and POW components. Crystals with tunable properties have been self-assembled and the structure is determined. Heterologous species have been organized by DNA nanoconstructs. Nanomechanical devices have been assembled from branched DNA, including shapeshifters and walkers, and they've been combined on an origami surface to produce a nanoscale assembly line, and then a crystal to produce a 3D device. And DNA nanotech has now been extended to the micron scale. I know there are a lot of students here, and a lot of students will often ask me, well, what should I study if I want to do DNA nanotech? Well, some answers are obvious. Math, physics, chemistry, crystallography, physical chemistry, organic chemistry, biochemistry, and organic chemistry, and perhaps some other branches of the sciences. But that's not all. If you look at this, you recognize this is just like that first slide I showed you about how do you make things for DNA nanotech. And what you can see, this is from the floor of the Pergamon Museum in Berlin. And you can see it's basically just 4 different branch-shaped junctions made out of DNA, it looks like. because it's a double helix in each direction, red and blue, got the right polarity. And so, and it's 2,000 years old. So in addition to everything else, you shouldn't forget art. There are a lot of ideas in art, and that can help you with what you want to do a lot. It certainly helped me. People ask me what's next, going maybe from there to there. We don't really know. I don't know at all, but right now, but I don't have to know. There are over 350 laboratories, about 375 or 380 now, engaged in structural DNA nanotech, so I don't have to know. The field is running on its own, and I could be hit by a truck, it wouldn't matter. This is a slide lent to me by Henrik Dietz. It's about 5 years out of date, but it does show the way the field's citations have been growing over the years. Finally, I have to thank the people who have paid for the work over the years. They didn't always know they were paying for it, which is why some of them terminated me at certain points. But nevertheless, the work is there. And at the bottom here is my web page. It's a couple of years out of date. I haven't got all the papers on there, but everything else is pretty well there. And you can certainly reach me and contact me through that. So thank you very much. I'll be happy to answer any questions if any are forthcoming. We want to put some lights on the audience, because I can't see if there's anybody asking a question or not. Just have to shout, I guess. What?
We have a mic set up there for questions. People will be coming.
Okay, oh, great. Okay, so if anybody has a question, just come over to the mic that he says is set up there for questions, and we'll try to answer them.
Hello. Any questions you can come here and ask? Anyone? You can come up here and ask. Excuse me? Come from this side.
Yes.
Thank you, sir, for giving me this opportunity. Sir, my question is, you have taken the nanotechnology DNA from nanoscale to the micron scale, right, sir? Sir, my question is what is the use of it? Means what kind of it will support in future or right now? Means in what kind of technology we can use it?
Well, I mean, I gave a list of applications early on. Not necessarily on the microscale, but there are a lot of uses for colloids on the microscale, and that's not my long suit. But there are a lot of people who have been trying to do this type of thing for a lot of years, and we finally succeeded in doing it. And I should point out that the control of chirality, which we showed there, that's like the hardest thing to do. We can also control angles, we can control associations, and so forth. Basically, if you just look on the web and Google beads, and you get all these different colors and arrangements of beads, we can now make almost any of those and use them for building blocks to make bigger things on that scale. So there are many utilities on that scale that exist there, but that's not what we do. We design the DNA.
Thank you, sir. Sir, one more question. Sir, this structural DNA biology means there are quadruplexes, 6 DNA strands are getting together, forming one piece of DNA molecule. And is there any function in the genome of any living cell, like bacterial or any?
Not that we know of. This is chemistry. It's not biology that we know about.
Okay. Thank you, sir.
Thank you.
Hello, sir. So this question might seem a bit naive because this is the first time I'm knowing about this, but you talked about the origami arrays. So it seemed like you said that they're like sort of like coordinates, so you can sort of plug things in. So I imagine that this can be used to probably create like very, very complex and large
Patterns, yes.
patterns and structures. So has this been done, and like how is it even useful?
That's in the works. Not much in my lab because we only rarely work with origami.
Okay.
But other labs are very heavily invested in that technology.
Thank you, sir.
Sure.
Are there any more questions? Thanks.
Okay. Well, thank you very much then.